That's why I said it.
Yes, sir.
JP sir.
Shall we start, sir? And only.
Yes. Bhagesh sir joined with us. Sir, can we start, sir?
Sure. Yeah.
Yeah, please. Welcome, sir. Dr. Fernandez. Fernandez, sir.
Yes.
You can proceed. As our chief guest of this session and convener of this lecture workshop, Dr. Bhagesh sir joined with us. We wholeheartedly welcome you, sir. We can start our technical session by the moderator of Dr. M. Fernandez Durai, Assistant Professor of Department of Biochemistry. Yes, Dr. Fernandez Durai.
So very good afternoon to everyone present here. Very good afternoon, sir. So in the name of Almighty, I welcome you all for this afternoon session. So it's my privilege and proud to introduce our today's resource person, Dr. D.J. Bacteria, sir, the NSI Honorable Scientist and Chairman for Center for National Biological Resources and Community Development. So, sir's educational qualification is MSc in Agriculture and a PhD, University of Agricultural Science, Bangalore. Sir completed his post PDF at New Zealand, Australia, and UK and USA. Sir received so many fellowships like a Fellow of Indian National Science Academy and the National Academy of Science, National Academy of Agricultural Science, the National Academy of Biological Science, the Society of Agriculture Biotechnologists, Indian Phytopathological Society, and Association of Microbiologists India. Sir received so many awards at national level, like S.R. Vyas Memorial Award, Show Me Memorial Award, Dr. G. Rangaswamy Memorial Award, Professor T.S. Sadasivan Lecturer Award, Dr. V. Agnihotru Memorial Lecturer Award, Dr. R.Y. Roy Memorial Lecturer Award, Dr. Natarajan Memorial Lecturer Award, and SARS recognized award by NABS and a recipient of a Distinguished Asian Microbiologist Award at 2019 from Asian Mycological Association and several Lifetime Achievement Award. SARS published 418 research papers and 112 review articles and book chapters, and SARS published 11 books and 14 popular articles. SAR has acted as a principal investigator of 36 research projects in microbial inoculant for sustainable agriculture. SAR has currently acted as a principal investigator and a co-PI of 3 research projects. And SAR as acting as advisory committee member of ICAR, DST, DBT, CSIR, and ICFRE, UGC, NBA, Rubber Board, and Spices Board, IFS Sweden, and AICR Australia, and FAO, and UNIP, and GE. And also, SAR taught microbiology for UG and PG courses past 15 years, and SAR has guided 40 MSc students and MPhil students and 25 PhD students. So we feel proud to have this session, sir. So once again, I welcome you all and welcome you, sir. Thank you.
Much. And only thing is, as Dr. Nias has told, that I'm not feeling well. I underwent a surgery and still recovering, so I'm not able to deliver the talk as such. I have already sent a recorded of my 2 talks, so I think you can project that. At the end, I'll be happy to answer the questions, whatever is raised by the students and staff. So you can go ahead and make the things. Only I would like to say that I had the opportunity of visiting your college earlier and really enjoyed the visiting your college and the interaction with the staff and students of the college. It is a wonderful place. I really liked it. So with this introduction, I will just say straight away, you can go with the projection of the first talk on Arbuscular Mycorrhizal Fungi for sustainable agriculture.
Sure, sir. Sure, sir. Thanks for your. We are highly fortunate to have with you. As per your word, we have the interaction session with you in a physical mode. Thank you, sir. We have prepared for this play that recorded video lecture. Hopefully, Dr. Jayaprakash will proceed with the presentation of the SARS recorded lecture.
Okay, sir. I will do it now. Okay.
Especially was interested in pathogenic fungi, those which cause disease in Today I'm going to talk on arbuscular mycorrhizal fungi and their role in sustainable agriculture, horticulture, and forestry. And so, first of all, we should know what the word mycorrhiza stands for. I'm sure that most of you know what the word mycorrhiza stands for. We know that myco stands for fungi, rhiza stands for root. So it is an association between plant roots and certain fungi.
Yeah, that's right.
And going
Kindly
back to
rotate
the
that,
history of
screen, sir,
this
please.
group of organisms, and
That's fine,
it was
that's
a
fine.
German botanist who coined this word mycorrhiza. And he was a botanist but interested in fungi, especially forest fungi. So he used to go to the forest, collect the fungi, try to identify them, and especially was interested in pathogenic fungi, those which cause disease in forestry species. So he used to collect the pathogens, isolate the pathogen, multiply it in the laboratory. Then he used to inoculate healthy seedlings with the pathogen which he has isolated to prove the postulates, and then prove that it gets diseased. The healthy seedlings become diseased. That is what the main type of work he was doing. So once he found that during a forest trip, the roots of pine seedlings were covered with certain fungi. So he thought it must be a pathogen. So he brought the samples, isolated the fungus, Ectomycorrhiza. It was an Ectomycorrhiza, so we can isolate it. It can grow on laboratory medium. So he grew this fungus, and then he wanted to prove the postulates. So he inoculated healthy pine seedlings and waiting that they would get diseased. But the opposite happened. What happened? The inoculated seedlings started growing better instead of becoming diseased. So he was surprised. He repeated it 2-3 times, and then every time it happened. And then he thought the fungus which he is inoculating must be a symbiotic fungus which helps plant growth. And that is how he coined the term mycorrhiza, which means an association between plant roots and certain fungi. And he also added one more word to that, the association is symbiotic. That is a very important statement he made. So that was in the year 1884, and that happened in Germany. And after that, nobody worked on this particular group of organisms or the mycorrhizal fungi. And it was late in 1950s, a student from University of London, when she registered for PhD degree, she was asked to select the thesis problem. So usually in our country, the guide gives the problem to the student. But in Western countries, it is the student who has to select the thesis problem. So when she was asked to select the thesis problem, she saw the word mycorrhiza was coined and described by the German botanist, and later nobody worked on it. So she decided to work on this association. So her PhD thesis problem was on mycorrhizal association in apple, which is a very important fruit crop in England. So she completed her PhD. After completing her PhD, she joined the Rothamsted Experimental Station in England, which is considered to be the mecca for the agricultural scientist. Everybody wanted to go and work in this particular station because it was so famous for agricultural research. And she joined there as a scientist, and she started working on mycorrhizal fungi. In addition to her research on mycorrhizal fungi, one great thing she did, she invited students or young staff from any part of the world to come and work in her lab on mycorrhizal symbiosis. And she also gave them the fellowship. So that attracted people to go to her lab and work on mycorrhizal symbiosis, so that when they go back to their own country, they can initiate work on mycorrhizal fungi. Okay, that is how the Mycorrhizal Association or the mycorrhizal symbiosis work spread throughout the world. And that is the reason why we call Dr. Barbara Mosse as the mother of mycorrhiza research. Many groups of science have fathers, but this group has a mother. And when we organized the International Conference on Mycorrhiza in Bangalore a few years back, we invited Dr. Barbara Mosse to be the chief guest, and she gave the keynote address. Now, we are in 2020, and now we know there are 4 major types of mycorrhizal association. Okay, the Ectomycorrhizal Association, which is commonly found in temperate forest tree species like pine, for example. And the fungi associated are all mostly basidiomycetous fungi. Okay, so in our country, we can see the Ectomycorrhizal Association in the Himalayan region, and that the temperate region, the very cold place. The second one is the vesicular arbuscular mycorrhizal fungi, which is very common in agricultural crop plants. So I will not go into it right now because I am going to talk in detail about this little later. And the next category is Ericoid mycorrhizal fungi. And as the name itself suggests, that it is this particular group of fungi are specific to plants belonging to the family Ericaceae, like azalea, rhododendron, blueberries, cranberries, and so on. Again, temperate. And so in our country, we have some rhododendrons and azaleas growing in the Himalayan region, and we do not grow blueberry or cranberry in our country. So as a result, we can see azaleas and rhododendron colonized by Ericoid mycorrhizal fungi in the Himalayan region. The fourth category is the Archid mycorrhizal fungi. As the name suggests again, that it is very specific to orchids, and these mycorrhizal fungi are obligate symbionts. That means orchids cannot grow without the association of this mycorrhizal fungi. So with the protocarms of the orchids, when they start growing, they must be infected by the mycorrhizal fungus. If they are not infected by the mycorrhizal fungus, they do not develop into a plant. So to that extent, orchids are dependent on this group of mycorrhizal fungi. So these are the 4 main types. Coming to the second type, the vesicular arbuscular mycorrhizal fungi. These are the ones which are very, very important in tropical countries like ours. And it is very important in crops, agriculturally important crop plants. That is, when I say agriculturally important, it includes plants important in horticulture and also tropical forestry. And I have to add one more thing here. Earlier, they were called as vesicular arbuscular mycorrhizal fungi. I will tell you the reason. Now they are called as only arbuscular mycorrhizal fungi. The word vesicular has been deleted. I'll tell you the reason little later. So here you can see here in the top we can see the non-mycorrhizal root, a transverse section of a non-mycorrhizal root where we have the epidermis and then endodermis, and in between we have the cortex. And here to the left side we can see a transverse section of a root colonized by the arbuscular mycorrhizal fungi. The fungus lives in soil, and in the soil it produces very large spores called as extramatricular chlamydospores. And the size of the spore varies with the species. And in some species, it can be as big as 450 micrometers. So one can be able to see the spore without the microscope. So the spores can be that big. And the hypha in the soil, it can penetrate the epidermis and then go to the cortex, and it ramifies in the cortex region. It never goes and penetrates the endodermis. So it is restricted to the cortex region, and then it produces structures called as vesicles, which are globose to round and ovoid, and also highly branched hyphal structures called as arbuscles. Since they produced these two structures, vesicles and arbuscles, they were called as vesicular arbuscular mycorrhizal fungi initially. Later, it was found few genera do not produce vesicles, whereas all of them produce arbuscles. So as a result, people deleted the word vesicular, and now it is called as arbuscular. So if you see the literature, scan through the literature, sometime before 1980s or 1990s, you will see that they are called as vesicular arbuscular. Now they are called as arbuscular mycorrhizal fungi. So I am sure they are ubiquitous. These fungi are present everywhere. They are present in almost every part of the world. And so if you just go outside in your campus, the university campus, try to bring the root sample and then stain it with a particular stain called trypan blue, I am 100% sure that the plant will be colonized by the mycorrhizal fungus. So you can see these spores, the vesicles which are inside the root. So you can see how many vesicles are inside the root. So what we see as a root is just the not root. It is colonized with the fungus, and it is filled with these structures called vesicles, and also the arbuscules. Arbuscules are highly branched hyphal structures, usually dichotomously branched, but they are very short-lived. So it's difficult to see the fine structure dichotomous branching very seriously or critically, because they undergo regeneration, disintegration in 4 to 5 days. And so they just disappear in 4 to 5 days. And that is the reason we are not able to see this hyphal branching very, very clearly. So what we are seeing here are the arbuscules. And all these fungi, the arbuscular mycorrhizal fungi, belong to the phylum Glomeromycota, which has 3 classes, 5 orders, 14 families, 26 genera, and 230 described species. Of course, this is by one particular taxonomist. It varies with the various taxonomists. Some people say there is only one class, so it varies with the taxonomist how they look like. But this is one of the mostly accepted taxonomic classification. So I said there are 26 genera of fungi described so far. Of these 26 genera of fungi, 4 of them are more frequently occurring. That means if you just go out and take the soil sample anywhere in the world and try to bring it to the lab and do the analysis by a very simple technique called as wet sieving and decantation technique, and then see through the microscope, and you will see these 4 are the most commonly occurring genera. And the 4 commonly occurring genera are these. A is Glomus, B is Gygospora, C is Aculospora, and D is Enterophospora. So these are the 4 commonly occurring fungi. And if you ask the question, of these 4, which is the most common? The answer is Glomus. A is the one which occurs very commonly in almost all the soils in the world. So again, if you just go out in the university campus, take a soil sample, bring it to the lab, do the wet sieving, and then see the sample under the microscope, I am 100% sure you will definitely come across Glomus. They are so common in Indian soils also. And these fungi are all obligate symbionts. So what I mean by obligate symbionts? we cannot culture them in a culture medium like other fungi. We cannot, the fungus cannot be grown on a potato dextrose agar or Zipex medium or Richards medium in the laboratory. They need the roots of a living plant to grow and multiply. So this is the way the fungi are maintained in a glass house with a suitable grass as the host. So each row is a particular species. This is how we maintain the fungi in our lab. And so this is, one can maintain it and use it as and when we require it. And this is a very old slide, in fact, of 1970s. And again, there's a story which I want to tell, and that is the reason why I'm retaining this particular slide. In 1975 or 76, I was offered a Fulbright fellowship by United States government to go and work in one of the labs there for 1 year. So that was University of California, Davis campus, and I was very lucky to get the mentor, Professor Mortimer Starr, who was the editor-in-chief of the annual reviews of microbiology. So when I went and then started working, the research project in which I was supposed to work was transferring a nitrogen fixing gene from a nitrogen fixing bacterium to a saprophytic bacterium and see whether it becomes a nitrogen fixer. That was the main objective of the project. So when I started my work, the next lab was working on mycorrhizal fungi. So I used to peep into the next lab. Somehow I felt what the next lab is doing is more interesting than what I started working. So I went to Professor Starr and asked, can I shift to the next lab? Then he said, you have come on a Fulbright fellowship. The head office is in Washington, DC. So you have to call the head office people. And if they have no objection and the other professor has no objection, I have no objection. So I called Washington, DC. The lady who was in charge of the program, she took the phone. And then she said, if both the professors have no objection, we have no objection, you can shift. Then the other professor, the mycorrhizal professor, had no objection. So I shifted to the next lab working on mycorrhiza. So that is a starting point when I started working on mycorrhizal fungi. That was in University of California. So my fellowship was to end on 31st December. And sometime in October, the Professor John Mengi, with whom I was associated working on mycorrhiza, he came and then said, we can take you, continue as a scholar for 5 more years on a contract basis as a scientist, if you like to continue with us. So I was very much tempted, thought I should continue in such a good lab. So I talked to my family, and both my wife as well as my mother said, they come back. And I talked to the Indian friends who were there in the campus, and all of them said I should come back, go back to India. The reason they gave was very, very convincing. They said they are all sticking on there because they never had a permanent job in India or there. So they said, you are already having an associate professor job in a government university, so you should go back. So I decided and told the Fulbright office I am going back. So some people said I am a fool and all that, usual stories. And then anyway, I decided to come back. So when I came back in January, I asked the postgraduate students to go and collect the roots of some of the cultivated plants grown in the campus. Being an agricultural university, we grow lots of crop plants. So they collected and we stained and looked for Mycorrhizal Association. So they were all beautifully colonized by mycorrhizal fungi. And it so happened very little work was done in India on mycorrhizal fungi. So we could start from a scratch. So many of these things, these are the very early slides taken in 1970s, and some of them became the world first report because The Western countries, they don't grow the crops what we grow. Just to give example, ragi they don't grow, for example, and clove, cinnamon, many crops what we grow here, they don't grow. So as a result, many of the world first report came from Bangalore, our university. And I also told them to go and bring some soil sample, and we analyzed the soil sample, and it had plenty of mycorrhizal spores. So our soils are rich in mycorrhizal fungi, and the plants are colonized by the mycorrhizal fungi. But what will happen when we inoculate with an efficient mycorrhizal fungus? Because when I came back from US, I asked Professor Menke to give me the best mycorrhizal fungus they have, and he was very kind enough to give the best culture what he had. And that happened to be the culture, the mycorrhizal fungus, which he used to give it to the farmers who are growing oranges and avocados. So that best fungus he gave me. So I brought it here, and my first experiment was done with that particular fungus. So this was done in Bengal gram, and we used the autoclaved soil here. You can see the difference, inoculated versus uninoculated, such a big difference. And we felt this big difference we got because we used sterilized soil where we have killed all the native endophytes. So we thought we should work with the natural soil which has the native mycorrhizal fungi. So next few slides will show the response done in natural soil. So this is in case of groundnut, uninoculated versus inoculated. You can see the difference. There is an improved plant growth even though our soil has the native endophytes, and we have inoculated with an efficient mycorrhizal fungus from US, but it is responding very well to that fungus. And this is in case of cardamom. Again, the same story. You can see the difference between the uninoculated and inoculated. And this is a forestry seedling. This is neem. You can see the difference between inoculated and uninoculated. So we have done it with several crop plants, and I have just included 3 slides. One is, which is agriculturally important groundnut, horticulturally important cardamom, and neem, which is an important forestry species. Okay, the next question is, how does it make the plants grow better? Okay, so in this particular slide, if we take, let us take the example of cotton. You can see the difference in the dry weight between the uninoculated and inoculated. Such a big difference because of inoculation with the particular fungus Glomus fasciculatum. And again, you can see the difference in the phosphorus uptake, zinc uptake, manganese uptake. Very, very clearly showed these fungi help in the uptake of certain nutrients. Okay. Then the question comes, which type of nutrients? Here we have to understand some basics of soil science. What we have to know is we know that nitrogen, phosphorus, potassium are the 3 major plant nutrients, and some of the nutrients move very freely in soil solution and come to the root hair zone. We know, all of us know that root hair zone is the structure which takes the nutrients from soil and supplies it to the plant. So some nutrients like nitrogen, potassium, they move very freely in soil solution, and they are called as mobile nutrients, or nutrients moving through mass flow, and they come to the root hair region and makes it available for plant growth. But there are some nutrients which are not mobile in soil solution, which we call them as immobile nutrients or diffusion limited nutrients. Example is phosphorus, which is a major plant nutrient. It doesn't move freely in soil solution. In such a situation, what happens? So we have a plant root, and these are the root hairs, and that is a structure through which the plant takes the nutrients. All that available phosphorus in the root hair region is taken by the plant. And once it has taken, though there is phosphorus away from the root hair region, it is an immobile nutrient. It is not able to move to the root hair. So there is a phosphorus depletion zone where there is no available phosphorus in the root hair region. So here the mycorrhizal fungus plays the most important role. We can see the length of the mycorrhizal fungus, which is several times longer compared to the root hair. So this hypha scavenges a larger volume of soil, takes the phosphorus from soil solution and pumps it directly into the host plant, thereby short-circuiting the nutrient. And this is one of the major ways by which mycorrhizal fungi help plant growth. This is true not only with phosphorus, but other immobile nutrients like zinc, copper, etc. Okay. Being a microbiology lab, we were interested in studying the interaction between the mycorrhizal fungus and the nitrogen fixing bacterium. And again, we used a symbiotic nitrogen fixing bacterium, Rhizobium japonicum, and this was a field experiment that was done in soybean. And again, this happened to be one of the very first field experiment done with mycorrhizal fungi. So to conclude, what we got from this result, what we got from this experiment, and what we found was there is a synergistic interaction between the mycorrhizal fungus and the rhizobium. What I mean by this, the mycorrhizal fungus increased nodulation and nitrogen fixation by rhizobium, okay, and rhizobium increased colonization and pea uptake by mycorrhizal fungus. So the interaction was synergistic. So what happens when the dual inoculation with both the organisms is much better in improving growth? You can see the dry weight of the plant, it is much higher compared to single inoculation with either mycorrhiza or with rhizobium. Dual inoculation has got a very high dry weight. The same thing, being a field experiment, looked into the yield of the crop, so which is much higher, suggesting that the interaction between the two organisms is synergistic, and it improves not only plant growth, but it also increases the yield of the crop plant. Okay. And we repeated this with a free-living nitrogen-fixing organism, Astrobacter, the exactly same results. There was a synergistic interaction between the two. Dual inoculation improved plant growth much better than the single inoculation. The same thing we repeated with a phosphate solubilizing organism also. And here I have to tell that there was a controversy amongst the mycorrhiza workers, that about 50% of the mycorrhiza workers thought that the mycorrhizal fungi can solubilize insoluble form of phosphorus in soil, because in soil most of the phosphorus remains as an insoluble form, and so it has to be converted to a soluble form which the plant roots can take. So some of the mycorrhiza workers believe that the mycorrhizal fungus can solubilize unavailable form of phosphorus, but another 50% of the mycorrhiza workers did not believe this. Okay, so we did one experiment using P32 labeled tricalcium phosphate, which gave very conclusive results that the mycorrhizal fungi do not solubilize unavailable form of inorganic P that is present in soil. So that means it only helps in the uptake of available phosphorus. So, but we have certain phosphate solubilizing microorganisms, both fungi and bacteria. So their job is they solubilize unavailable form of P to an available form. How do they do that? It is mainly through the production of organic acids, oxalic, citric, and so on. So they produce this organic acids and convert the inorganic form and then convert them to a soluble form, H2PO4 ions, which the plant roots can take. So when we inoculate with the phosphate solubilizing organism and the mycorrhizal fungus, again there was a synergistic interaction. Dual inoculation improving plant growth much better compared to single inoculation. In this slide, the same thing in a diagrammatic way we are telling say some farmers will be using rock phosphate. Okay, rock phosphate is an insoluble form of phosphorus, and so when the farmer has inoculated with a phosphate solubilizing organism, it produces organic acids and releases the available PO4 form, and that PO4 is taken by the mycorrhizal fungus. So there is a synergistic interaction between the phosphate solubilizing organism and the mycorrhizal fungus. Another thing which we looked into is the interaction between a plant pathogen and the mycorrhizal fungus. This is in groundnut. You can see the groundnut plant, it is diseased with the pathogen Sclerotium ralphsi. You can see very badly diseased plant. Here we have introduced the same propagules of the pathogen plus the mycorrhizal fungus. You can see the difference between the two, which very clearly show that the mycorrhizal fungi helps to alleviate the severity of the disease caused by the pathogen. And we have worked out the mechanism by which it helps. They protects the plants, and the mycorrhizal roots have a higher concentration of orthodihydroxyphenols, which inhibit the germination of the pathogen, and thereby it protects the plants against the pathogen. And in and around Bangalore, we have the root knot nematode problem, very serious in cerealaceous crops. And this is tomato root, and you can see that big galls, tertiary galls in tomato roots formed by the nematode Melatogain incognita. And here to the right side, again tomato root, we have introduced the same number of larvae, nematode larvae, plus the mycorrhizal fungus, and it produced very small primary galls. So we were wondering how to interpret this result. So discussing with the nematologists and going through the literature, we could finally come to some conclusion. That means the primary galls are formed even when we introduce the mycorrhizal fungus. That means the mycorrhizal fungus is not preventing the penetration of the nematode larva into the root. That is the reason why they are forming the primary galls. But it is preventing the development of the nematode larva into adult. Thereby we are not getting the secondary and tertiary results. So this our nematology friends also accepted, and we scanning through literature, we found what prevents the development of nematode larva into adult. We could see that sulfur containing amino acids prevent the development of nematode larva into adult. And we looked into the roots of the mycorrhizal versus non-mycorrhizal root, and we could see that the mycorrhizal roots had a high concentration of sulfur containing amino acids, which clearly explained why the secondary and tertiary galls are not formed. Now, summing up the mechanism, how the mycorrhizal fungus helps plant growth, we can see that it helps in the nutrient uptake of especially the immobile nutrients like phosphorus, zinc, copper, etc. It has synergistic beneficial interaction with beneficial soil organisms like nitrogen fixers, phosphate solubilizers, PGPRs, and so on. And they protect the plants against soil-borne plant pathogens. And our recent studies have shown, again through an ICR project, very clearly that they protect the plants against water stress, that is drought. Okay, and they also produce growth promoting substances like indolactic acid, gibberellin-like substances, and tannin, and so on. And so they protect, it's a cumulative effect of all these which makes the plants grow better. And here I have to say something, what the mycorrhiza workers both from England as well as USA were telling, that mycorrhizal fungi are not host specific. It is true even today. What we mean by not host specific, you try to isolate the mycorrhizal fungus from wheat and try to inoculate tomato, it will colonize tomato plant. Take it from tomato and put it on banana, it will colonize banana. So that is why we call them as non-host specific. But this is one of the very interesting experiments done by one of my students. And here we have Lucina leucocephala as the host. The last one is control, which is not inoculated with the mycorrhizal fungus. And here he has inoculated with different mycorrhizal fungi. And what we could conclude from this, that this is the best mycorrhizal fungus for inoculating Lucina leucocephala. So we introduced a new hypothesis. Though mycorrhizal fungi are not host specific, they have host preference. That means for a particular host, there is always a best fungal partner which can enhance its growth to the maximum extent. So this particular hypothesis of host preference was not accepted by the British as well as the American scientists to begin with. So we have to show it in many hosts. This is again in cashew. We said the last one is the best mycorrhizal fungus for inoculating cashew. And this is in case of citrus rootstock. And the third one from left is the best mycorrhizal fungus for inoculating citrus. So with this, I think they were all convinced. The United States Department of Agriculture gave us a big project. The money came in US dollars. They said, if your hypothesis of host preference is correct, we are giving you this 5-year project. At the end of the project, what we want is one mycorrhizal fungus which can be used by the forest nurserymen for inoculating Lucina leucocephala, which was considered as a wonder tree few decades back. And this is the type of result we got at the end of 5th year. And there are 4 different cultivars of Lucina leucocephala grown in Karnataka state. And we selected the best mycorrhizal fungus, which will improve the growth of all the 4 cultivars. The reason is, suppose one farmer or one forest nurseryman asks, I want the culture for inoculating Lucina. We have to give one culture. We can't be giving different culture for different varieties or cultivars. So that is how this particular species, Glomus mossae, proved to be the best for improving the growth of all the cultivars grown in Karnataka state. You can see the difference in the root weight also, the structure. These are the 4 cultivars, K8, K28 and so on, where Lucina, which is grown here. And in agricultural universities, we have Krishi Mela, where the farmers come and the professors and the scientists, we have to face the farmers and tell them what the recent research work we have done and explain to them. So when I explained to them the mycorrhizal work on Lucina and told the farmers that Glomus mossae is the best mycorrhizal fungus, one farmer stood up and asked a question. He said, which is the next best fungus? And I said, it is Gygospora margarita is the next best fungus. Then he asked the next question, what will happen if I mix these 2 fungi and inoculate? And I had no answer. I told him, please come next year. I will do this experiment and give you the answer. And we did this experiment and there was no adverse effect by mixing both the organisms. And it improved the growth of Lucina leucocephala. And when this work on Lucina was going on, the Director General from ICFRE, the Indian Council for Forestry Research and Education from Dehradun, happened to visit the university. And he saw these plants, Lucina leucocephala plants, and he said, is your mycorrhizal fungus bringing such much difference in plant growth? I said, sir, that's the only inoculation. We have done nothing else. So he was so much surprised, as I said, we are giving a big project. So what I want is the best mycorrhizal fungus for inoculating various forest tree species grown in our country. So another project came, and this is what the difference we could see in case of Tectona grandis. And we have developed the best mycorrhizal fungus for various forestry species. Here I have listed them: Acacia nilotica, Auriculaformis, Holosaricea, Albizia lebec, Azadi recta indica, Casuarina, Dalbergia, Tamarindus indica, Tectona grandis, and so on. So when this work on forestry species was going on, the horticulture people came and they said, we want the best mycorrhizal fungus for inoculating various horticultural crop plants. So another project was given, so we could screen and select the best mycorrhizal fungus for various vegetable crops and plantation crops like coffee, cardamom, and even flowering plants like marigold, chrysanthemum, and plants like rosemary, and so on. And at that time, looking at these results, the Medicinal Plants Board came and they gave with another project. They said, we want the best mycorrhizal fungus for inoculating various medicinal and aromatic crop plants. Again, we have screened and developed the various best mycorrhizal fungus for inoculating various medicinal and aromatic crop plants. And here, having selected the best mycorrhizal fungus for various crop plants, the next question, what is the practical application? Of course, it will improve plant growth. Apart from that, is there any other important application? So to answer this, we developed these phosphate response curves. You can see that in x-axis, we have increasing phosphatic fertilizer, and the y-axis, we have the increase in the plant biomass. And here we have the non-mycorrhizal plant and versus the mycorrhizal plant. And to produce this amount of biomass, the amount of phosphatic fertilizer the non-mycorrhizal plant requires is, let us say, 70 kg. And to produce the same amount of biomass, the mycorrhizal plant will require, let us say, 50 kg. That means through mycorrhizal inoculation, we are saving 20 kg of phosphatic fertilizer. So this is the one which really caught the attention of the research scientists and also the funding agencies. This we have shown in many crop plants as field experiments. I have just included one, the chili crop, and the recommended phosphatic fertilizer for chili is 75 kg P per hectare. And the uninoculated plant, given the recommended level of phosphatic fertilizer, yield is 1.81 kg per plot. And this different mycorrhizal fungi, and this was the best, and you can see 50% of the recommended P. Okay, that we have to remember, 50% of the recommended P, and it gave even slightly more yield compared to recommended P, which very, very clearly showed 50% of the phosphatic fertilizer can be reduced through inoculation with an efficient mycorrhizal fungus. This we are shown with various vegetable crops, and so the next thing I would like to say is about the high-tech agriculture that is being followed now. I am taking the example of Bangalore again. The farmers around Bangalore, they never raise their own seedlings. They just go to a nurseryman, buy the seedlings and plant it in the field. So the nurseryman raises in pro trays or the root trainers, as we have seen here. This is how the seedlings are raised. And here, you see, this is the close-up, and you can see here, this is a time where it's much easier to mix the mycorrhizal fungus along with the substrate and raise the seedlings. And the roots are colonized by the mycorrhizal fungus. The farmer buys the seedling from the nurseryman, which is already colonized by the mycorrhizal fungus, and he takes it and plants it right in the field. So it goes straight into the field. So this is a very simple technology which can be followed as a nursery technology. At this time, the Indian Council of Agricultural Research came with another problem. They said, in case of citrus rootstock, the rootstocks sit in the nursery for 24 months. So the seedlings remain in the nursery for 24 months. Why? Because the stem has to reach a thickness of the pencil thickness when it is ready for grafting. So it takes 24 months for the stem thickness to reach the pencil thickness. So they said, we are giving a project whether this can be reduced by inoculation with the mycorrhizal fungus. So this was done by another student, and here is the uninoculated plant, inoculated with different mycorrhizal fungi, the stem dia you can see, and this is the best mycorrhizal fungus. You can see the difference between the uninoculated and the inoculated. And they reach this particular diameter in 18 months instead of 24 months. Thereby, the plants can sit in the nursery only for 18 months instead of 24 months, saving 6 months nursery period. And it's a very common technology which is followed by the citriculturists. And this time, in and around Bangalore, we have lots of floriculture. So these floriculture people came to me and said, how will mycorrhizal fungi help? So this is, we have done it with several flowering plants, and I've just included one as chrysanthemum, uninoculated versus inoculated. You can see the increased number of flowers. But another important thing which I have to point out, these inoculated plants flowered 3 weeks early. The floriculturists were more interested in this, that they flowered 3 weeks early compared to the uninoculated plant. At this time, the post-harvest technology department in the university, the professor came and she said, can we keep these flowering, the vase, the mycorrhizal versus non-mycorrhizal flowers in the flower vase and see whether they last longer in the flower vase? I said, ma'am, try to do it. It will be interesting. She did it and came and said the mycorrhizal flowers remained in the vase 3 days more compared to uninoculated plants. Now, this has been done in different countries now, and they all reported that mycorrhizal plants remain in flowering vase 3 to 4 days more. And that's because of reduced amount of ethylene that is produced in mycorrhizal plants. And here again, ICR came with another problem. The problem is cashew growers raise the seedlings in polybags, and then after they reach their particular height, and they plant it in the field. So what they noticed was 40% mortality. If they plant 100 seedlings, 40 of them die. That means mainly because of transplant shock of cashew. So ICR came to us and said whether the mycorrhiza can help us. Again came with another project, and we did this work. And here is the result of after 3 years. And you can see when they were inoculated with these two fungi, Acalospora leavis and Glomus etonicatum, the survival was 100% in the field. And in contrast to uninoculated, the survival was only 63%. That means the mycorrhizal fungi help the plant seedlings to overcome the transplant shock. Again, a common practice which is followed by the cashew growers. At this time, the tissue culture people came, and they came whether the mycorrhizal fungi can help in the tissue culture plants. At the time of hardening, we have added the mycorrhizal fungus. And so you can see the difference in the plant growth. And this is again a tissue culture plant of Ficus benjamina, which is commonly grown in between the double road in Bangalore because it can resist pollution. And so you can see the last one here. This is inoculated with only mycorrhizal fungus. And here the other last, we have inoculated with a consortium, the mycorrhizal fungus plus 2 more organisms, Trichoderma harzianum and Bacillus coagulans. So this gave us a clue that mycorrhizal consortium, microbial consortium, is better than inoculating with the single mycorrhizal fungus. So later on, we started concentrating on a consortium consisting of a mycorrhizal fungus plus PGPRs and biocontrol agents. So here this is in case of Acacia auriculaeformis uninoculated and versus microbial consortia. This is another DBT project which we completed recently, where there is a problem in tomato, wilt disease complex. As the name itself suggests, it's a complex disease. It is caused by 3 pathogens, a fungal pathogen, a bacterial pathogen, and a nematode pathogen. So it's not that easy to control because 3 kinds of pathogens are involved. And so we developed a microbial consortium consisting of a mycorrhizal fungus, a bacterial Pseudomonas fluorescens, and another fungus, Bacillomyces lilacines, which will take care of the nematodes. And this consortium worked very good in raising healthy seedlings versus the uninoculated seedlings. And some of the farmers may raise it in raised nursery beds. This is again the wilt disease complex caused by 3 pathogens. You can see capsicum uninoculated versus inoculated, which we can very clearly see the difference in growth of the inoculated seedlings. And we had another student who, I'm sorry, before that I would say this is again a response to inoculation with the Microbial Consortium. This is in case of patchouli. Patchouli is an aromatic cropland which is grown for the perfume, as the scent is extracted from this. And so this plant smells so good. So when the student, when she was working with this particular plant, she used to harvest the plant and used to shade dry in the laboratory. The whole building, I'm not talking of microbiology department, the whole building used to smell so nice that everybody used to come and ask what you are doing in your lab. So that wonderful smell it has, and that is because of a particular oil, the oil content that is present. So here I just want you to see with the recommended level of NPK fertilizer, see the amount of oil the plant is producing is 2.92. We have reduced 50% of NPK, nitrogen and phosphorus with the Microbial Consortium. See the essential oil, 3.66. That is the oil which the industry is interested in to produce the perfume. So this is a thing which is going on a large scale in a particular district in Karnataka, which grows lots of patchouli. And another crop, medicinal crop, which was introduced with the buyback arrangement from pharmaceutical company is Coleus fusculi. It was introduced in Tamil Nadu, Andhra Pradesh and Karnataka with the buyback arrangement. So farmers were very happy and they were growing it. But what happened? Sad news. It was attacked by a wilt disease, Fusarium wilt. And to what extent? Disease index was 85%. The farmers came with tears and said, this is what is happening. And with the Mycorrhizal fungus, and this was a field experiment which we did in a place called Kollegal, which is a border between Tamil Nadu and Karnataka. And with the Mycorrhizal fungus alone, you can see the disease index was 68. With the consortium with Trichoderma, it came to 33. So Coleus fusculi growers now take this particular thing as a routine practice for the cultivation of the crop. Again, a DBT project was given to us to develop the mass production protocol of fungi, being obligate symbions. So by this, we have developed this particular protocol with the best host, best substrate, the best time to harvest and how to raise the seedlings and so on. And so now this is taken by 4 industries, 2 in Tamil Nadu, 1 in Gujarat and 1 in Andhra Pradesh. And also it has gone outside the country, 1 in Malaysia and 1 in Indonesia, I don't know. Then another inoculum placement experiment which we did, placing the inoculum 2 centimeters below, if it is a raised nursery bed, gives the best result. If it is a poly bag, placing the inoculum 6 centimeters below the seed gives you the best plant growth. Okay. And how much inoculum is required? And here again, initially we said 1 kg of mycorrhizal inoculum per meter square is required, but now we have come down. We have come down to 500 grams, gives you very, very good result. So at this time, another Krishi Mela came when we have to face the farmers. I tell you from my personal experience, it is much easier to face the scientists and students. It's much more difficult to face the farmers because they ask very intelligent questions. And in fact, I have developed some research projects based on the questions the farmers have raised. And so this Krishi Mela came, and one farmer said, we appreciate the work done on mycorrhiza, but I have a 10-year-old mulberry garden. So the plants are old, 10-year-old. Can I inoculate with mycorrhiza? And if I inoculate, will I get better yield of leaves? Because mulberry is known for leaves. And Again, we have not done such an experiment with old plants, grown up plants. So I told him, please come next year. I will do this experiment for you and give you the result. Fortunately, the Sericulture department of the university had a 10-year-old mulberry garden, and we inoculated with the mycorrhizal fungus in the root zone, and we found that the crop responded to inoculation and gave with the 50% of phosphatic fertilizer, the yield was same as that of 100% phosphatic fertilizer, which very, very clearly showed. We repeated this with papaya, grown-up papaya trees, grown-up clove, and we found that even in the grown-up plants, if the mycorrhizal fungus, selected mycorrhizal fungus, is introduced into the root zone, they get better growth and yield. And at this time, I have to introduce this particular slide. And you know, the planning commission was there earlier. Now we don't have planning commission. And one of the planning commission meetings, I was invited as a special invitee. And the reason they said, we heard so much about the mycorrhiza research that is going on, but what we want to know is about economics. We are not worried about the response and yield and all that. What we want is about the economics. I was a bit scared because of all bureaucrats in the, all IAS officers mainly in the planning commission. So we worked this particular slide. I took tomato crop, which with the research what we did with, the yield of uninoculated tomato crop given 100% phosphatic fertilizer, the farmer would get 9,000 kg yield per acre. And if he is reducing the phosphatic fertilizer by 50%, inoculating with the best mycorrhizal fungus for tomato, then he will get 10,400 kg. This is all we have collected through our field experiments. So that means he will get 1,400 kg extra yield. And the yield of tomato the cost of tomato varies in Bangalore. Sometimes you can't believe it comes to 2 rupees a kg. Sometimes it goes to 100 rupees a kg. So I have just taken 25 rupees as a average. And then here the farmer gets 1,400 kg extra yield. So multiply by 25 rupees, he gets 35,000 rupees more. And he has saved 50% of phosphatic fertilizer. And so totally, he gets 36,875 rupees per acre as profit. So this convinced the bureaucrats, the planning commission members, and that is why it was recommended as the first area of research and for the different funding agencies. And at that time, I was to retire from the university. And one of the DST meeting I was attending, I was as an expert committee member. So one of the DST person asked me, what are you going to do after retirement? I said, I'm going to continue in the university as emeritus professor. He said, that is fine. You continue with your teaching and research. But we want that whatever the good work you have been doing should go on a larger scale to the farming community. It should go to the society. So what he suggested was we should start a trust and then continue only research. So that is how we started this particular trust in addition to my emeritus professorship in the university. And that is what is Center for Natural Biological Resources and Community Development, with which I am attached now. And the first project was given to the trust. What is the first project given? And of course, they said the trust must be 3 years old to give a project by Government of India. But they said, since you are already a committee member, expert committee member, we will waive off this regulation. We will give you the project in the first year itself. But with a question mark, what the question they raised, your PhD students have shown such wonderful results with inoculation with mycorrhizal fungi. Maybe 100 uninoculated plants with 100 inoculated plants, they show such beautiful differences in growth. Suppose a forest nursery man raises thousands of seedlings, will he get the same response? So we want you to try this. So that is how that was the main object of the first project that was given to the trust. And you know the number of seedlings? They said 9,000 seedlings. I was so scared. 3 forestry species, each in 3,000 seedlings, 9,000 seedlings. And here is the person who was only one senior research fellow in the project. And I was wondering how this one person will manage 9,000 seedlings. And now he is associate professor. And the Karnataka Forest Department came to my rescue. They said, don't worry. You want 30 laborers, you want 40 laborers, we will give. But what we want is this technology, how it works. That's what we want to know. So they gave plenty of labor. So there was no problem in doing this research. And this is the result, what we got with 3,000 of each one of these seedlings. You can see Tectona grandis, uninoculated versus inoculated. Dalbergia sissu, uninoculated versus inoculated, and Acacia auriculaformis, which very, very clearly showed what my PhD students did was a wonderful work, which works even when thousands of seedlings are raised. So this is the result. And the whole thing was done in a place called Mandya, which is between Bangalore and Mysore. And there was a degraded forest in that area. And the forest people said, why not we plant these seedlings in the forest, degraded forest, and we will give you all the help, no problem, because such a slope, you know, one has to climb and then dig the hole and then plant. And finally, we could do it. And this is 1-year-old Tectona grandis. You can see how bad, when I say degraded forest, you keep the soil, how bad it is. And this is the type of difference we get in 1 year. And this is in case of Acacia auriculaformis, 1-year-old. Sorry. And this is Dalbergia sissu. Actually, I really want you to see this. This is the forest man standing in uninoculated trees. These are 52 months after planting. 52 months after planting. And these are the inoculated trees. And the same man is standing here. He has become small because the trees are very big. And inoculated trees were double the size of uninoculated trees. So this is the difference. And of course, the DST funded project, I invited Dr. Vinita Sharma. She came all the way from Delhi to see the results and said, this simple technology should go to all the other states in the country. I don't know when we are going to do it. I am busy with too many things. And so I hope somebody will do it. And we do organize training programs for the farmers, forest nurserymen, horticulture people who are interested in the microbial technology. And what we can conclude from what I have said so far, at the time of independence, there was shortage of food in our country. We were eagerly waiting for the ship to come from United States carrying wheat, because when wheat came, we can grind it, make rotis, and then eat it. So that was the situation. Thanks to Green Revolution, high-yielding varieties of croplands were introduced. We all know the work of ICR, work of Dr. Swaminathan. The high-yielding varieties were introduced. And we were very happy because we could get enough food which can feed the people. But after several decades now, we are worried about the environment. Okay, so for raising the high-yielding varieties, what were used? Heavy doses of fertilizers and pesticides were used. Now, we are very much worried about the environment. Why we started worrying about it? Because in many places, people saw that the nitrate level in the water, drinking water, is much higher than the recommended level. People have recorded some pesticides in the tender coconut water, which we just we buy and then drink, and they have found pesticide in that. So we are now concerned about use of heavy doses of fertilizers and pesticides. So what do we talk now? We talk of organic farming, we talk of sustainable agriculture, which suggests use of less chemicals and more biologicals. And you know, the government of India has already made one state completely organic. Sikkim is 100% organic. They cannot use any chemicals in agriculture. And so it will spread to other states slowly. And so when we think of other biologicals instead of chemicals, we can think of green manures, compost, biocontrol agents, biofertilizers, all these come into the picture. So here the mycorrhizal fungi and other microbial inoculants play a very, very important role. And another important thing is they are cost effective. They are much cheaper compared to a chemical fertilizer or a pesticide. They are environment friendly. They improve the soil health. And now again, all of us know FAO is giving so much of importance to soil health. Why? If the soil is healthy, the plants that are growing are healthy. And if the plants growing are healthy, animals and human beings in turn will be healthy. So these microbial inoculants, including mycorrhizal fungi, are boon to farmers, especially in a developing country like ours. So with this, I conclude and thank you once again. And if you have any questions, feel free to ask or put it in the board. Chat.
Yes, sir.
Yeah,
Participant, do you have any questions? You can ask now.
Yeah, can ask or put it in the chat box, maybe better. Yeah.
ஒரு இது வெபினார் ஒன்று அவங்க தான் குரூப்ல போட்டிருந்தாங்க மெக்ஸ் ஸ்டாஃப்ல நீ பார்க்கலாம்.
I see one question here. What does Arbuscular mycorrhizal fungi do? 1 hour after listening to this, you are asking this question. Okay. So now you know, I think, after listening, what they do for plant. How do they help in plant growth and protect the plants against soil-borne diseases? Okay, the next question is, how does it work for benefit farmers in the form of high yield? Again, it is mainly through the nutrition, especially the diffusion limited nutrients. When they say diffusion limited nutrients, one should understand some nutrients in soil move very freely in soil solution, which we call as nutrients which move through mass flow. There are some nutrients which do not move through freely in soil solution, which are called as diffusion limited nutrients. Phosphorus, zinc, copper are examples. Phosphorus is a major nutrient. So mycorrhizal fungi help in the uptake of such diffusion limited nutrients, protects the plants against soil-borne plant pathogens, produces plant growth promoting hormones like cytokinins, indolactic acid. So all these together, and also they help in the water uptake during drought situation. So all these help the plants to grow better. Another question, any harmful mycorrhiza to human? So far, no reports of such reports, anything harmful to mankind. What is the biggest challenge faced while working on fungi? The main problem is the mass production. Being obligate symbionts, it cannot be easily multiplied like Rhizobium or Ascobacter or Pseudomonas. So we have the problem, or like Trichoderma. We have to grow them along with the host plants. Of course, there is an alternate technique called root organ culture, where few species of fungi can be multiplied through root organ culture in the laboratory. So again, the drawback is we can produce a lot of spores, but to what extent their infectivity potential is sometimes very less. That is one problem. And again, all fungi cannot be produced through root organ culture. Another question is here. Is there any harmful impact to humans who take the mycorrhizal inoculated crop plants as food? So far, no. Okay. Then wonderful presentation, very clear cut. Okay. Is there any other questions also? You can ask. I'll be happy to answer.
Yes, sir.
Yes, sir.
No,
I think there are no more questions. We shall proceed to the technical session 4, sir.
okay.
Can we move on to next session, sir? If you
Okay.
permit,
Yeah.
can present with.
After a few minutes, is that?
One more question is there, sir, in chat box. Please respond. Please.
One more question I see here. Can mycorrhizal fungi survive without plants? If it is so, how long do they survive? They do not survive without the plants. The host is a must. Otherwise, they die. We have done so many experiments, leaving the land fallow, growing the non-mycorrhizal plants, and their population comes down in one season. About 3 months' time, the population comes down if there is no plant. Okay, we are starting the next session after about 3:40 or what, whenever you feel like.
Yes.
Yes, Dr. Fernandez Durai.
Yeah.
Sir is asking
The
whether we
session will be
are
continued
getting a
or-
break or we'll continue.
Anything is okay for me. Yeah, because I'm not
Or we can
talking.
proceed with next lecture, sir, without a break, because we can proceed with next lecture.
Okay.
Yeah.
Okay, sir. I'll share the video.
So, if for him it is which source in which plants grow. Okay. So, if for him it is very, very important. Whereas for a person coming from big cities like Delhi, Mumbai, Chennai, Bangalore, if a person, a lady or a man walks in and then his plant or sari becomes dirty with soil, what do we know? We want to get rid of the soil. So that is how a city person looks at soil. To a civil engineer, how does it look? So when he is starting construction of a building, first he digs the soil to take the foundation and he throws away the soil. That soil is not needed by him because just for the foundation's sake, he has taken the soil. And similarly, for a biologist, it is a fascinating habitat teeming with life. It has not millions, billions of organisms. Okay, so it is very, very important for a biologist. Soil is formed by the weathering of rocks. So this slide shows the soil profile. We know that soil is formed by the weathering of rocks. It may take hundreds, sometimes thousands of years to form a good soil. And when we see this here, we can see that is the basic rock. That is a hard horizon, the parent rock. It has to undergo degradation. So partially weathered rock is the sea horizon. And the top A and B are the soil formed by the weathering of the rocks. The B horizon is at the lower depth and A horizon is the top soil. So usually A horizon of the soil which we see is 0 to 20 centimeters, which supports plant growth. And it indicates a very high level of organic matter, helping plants to grow. The one deeper below that is the B horizon soil, ranging from 20 centimeters to 100 centimeters. And deeper rooted plants also go to this particular B horizon and they draw the nutrients. So A and B horizons are the ones which are important for helping plant growth. And a fertile soil is vital for human survival. And it is estimated that 99% of the world's food come from the plants which grow in the soil. And the animals also feed on the plants, and thereby we get our food from plants as well as animals. And life within the soil is hidden. We cannot see what is the life within the soil, living organisms. So we see only the ones which are above soil. So because the organisms below the soil are out of sight, many times they are also out of our mind. So soil biology or biodiversity is a study of biological systems occurring in soil. And it supports a variety of flora and fauna. Thus people call it that as a factory of life. United Nations declared 2010 as the International Year of Biodiversity. And the organisms in the soil contribute to 25% of the total biodiversity in this planet. So 25% of the biodiversity in the planet lives in soil. And we know that United Nations also declared the years 2011 to 2020, which we completed very recently, as the decade of biodiversity. And how can we classify soil organisms? So they are classified based on the body size. If they are between 1 to 100 micrometers, if including bacteria, fungi, we call them as microflora. If the size is between 5 to 120 micrometers, which includes microfauna, which includes protozoa, archaeozoa, etc. And the organisms are still bigger, 80 micrometers to 2 millimeters, including columbolans, nematodes. We call them as mesofauna. And if are still bigger, size ranging from 500 micrometers to 50 millimeters, which includes earthworms, termites, etc., we call them macrofauna. So this is how the soil organisms are classified as microflora, microfauna, mesofauna, and macrofauna. And inventories on soil microorganisms are limited compared to the organisms living above ground. So again, when we go to the soil organisms, if the organism is bigger in size, it has received more attention and more studies have been made. If they are smaller in size, less studies have been made. And so the microbes are very small in size. So that is how we know only about 3% of the microbes living in soil. And here the same thing, I have just given another table. When we talk of vascular plants, the estimated number of vascular plants are 300,000. But how many we know so far is 270,000. That means we know 90% of the plants living above ground. So when we talk of the below ground, when we talk of ants, we know only 58% of the plants, of the ants. Coming to termites, we know only 53% of the termites existing, supposed to be living in this soil. And coming to mites, we know only 3%, columbolans 27%, and only 1.3 of nematodes. And protozoa, we know about 7.5%. And bacteria and fungi, as I told, only 3% of the organisms existing in nature we know so far. So this chart just gives you the various soil biota, which includes macrofauna, mesofauna, microfauna, bacteria, fungi, algae, and the organisms which are smaller than bacteria, which includes mycoplasmas, viruses, viroids, and prions. I will go through each one of them very briefly. Let us start with macrofauna. Macrofauna includes earthworms, mollusks, insects, millipedes, earwigs, etc. What is the major role played by them in soil? They play the major role is organic matter breakdown. And they also improve soil structure aeration, and water infiltration. And they also predate on soil microorganisms, thereby they maintain the biological equilibrium in soil, which is very, very important. Just included few slides of macrofauna, which includes thrips, termites, ants, centipedes, millipedes, earthworms, and it also includes a variety of beetles. You can see variety of beetles here, and the one here is the dung beetle. I think people coming from villages must have seen that some beetles roll the cow dung and even human feces and take it to the nest. So all these come under macrofauna, and still bigger also are there, which are covered under this group. Spiders, slugs, snails also come under macrofauna. This is a very interesting picture which I could get. You can see that the earthworm in this man's hand. See the length of the earthworm. It is as tall as this man himself. So this has been found in Amazon forest in Brazil. So the name of the earthworm is Rhinodrilus prioli. And even in our country, in Western Ghats, we have very big earthworms. Sometimes people can mistake they are small snakes. So this is the earthworm which we see in Western Ghats, and it is named Dravida species. Coming to the next category, mesofauna, which includes mites, springtails, rotifers, nematodes, tardigrades. Of these, the one which has been studied mostly are the nematodes. And we have different kinds of nematodes, which are pathogenic, herbivorous, omnivorous, carnivorous, and so on. And we know nematodes are important plant pathogens causing diseases in plants. And there are certain non-pathogenic species of nematodes which regulate microbial population. So they maintain the equilibrium of the soil microflora. I just included some mesofauna here, which includes tardigrades, nematodes, rotifers, mites, and springtails. And coming to the next category of soil organism are the microfauna, which are smaller in size, which includes protozoa and archaeozoa. Archaeozoa are just like protozoa. Only difference is that they have no mitochondria. That's the only difference between protozoa and archaeozoa. And both of them are important predators of bacteria, algae. Thereby they regulate the microbial population in soil. Some of them are pathogens in drinking water. And we also know about the amoebic dysentery, and which Entamoeba histolytica, and that is present, that can contaminate drinking water and cause amoebiosis. Similarly, there is another protozoan, Giardia species, and which can cause giardiosis, which results in foul-smelling watery diarrhea in man. I've just included few slides, photographs of amoeba, flagellate, and ciliate, which come under microfauna. Coming to the next category of organisms found in soil are algae. Algae are all photosynthetic and aquatic. So they prefer a marshy environment. And what is the role they play is, they play a very important role in soil formation and soil aggregation by adding organic matter. Some of them fix atmospheric nitrogen. All of us are familiar with Nostoc and Abena, which fix atmospheric nitrogen. And they form symbiotic association with certain fungi. And this association is called lichens. And you can see them on the tree trunks or on the rocks, which play again very important role in weathering of rocks. The next group of organisms are fungi. It can be either microscopic, single cellular like yeast, or they can be big, which are visible to the naked eye like mushrooms and puffballs. So what is the role played by these fungi? They play a role in the decomposition of organic matter, soil aggregation, nutrient cycling, about various cycling which we have studied, phosphorus cycle, sulfur cycle, and so on. And they also play a very important role in the biocontrol of plant pathogens. And some of these fungi are edible. And we all go to the market and buy mushrooms and eat. So that is, some of them are edible. Some of them cause diseases in plant and animals. And we are very familiar with the diseases of plants and, and also animals. I'll just come to that a little later. And after this slide, we know that yeasts, which are single cellular, play a very important role in fermentation and the bakery, bringing out bakery products, penicillium, fungus, ectomycorrhizal fungus, mushrooms, and nematode trapping fungi, which can trap nematodes and kill them. So these are all examples of fungi. And we know that many of these fungi cause plant diseases and also animal and human diseases. Coming to the next category, bacteria. Bacteria in any soil contains plenty of bacteria. And here the bacteria are present not only in soil, but in the regular soil, but also in extreme environments. Organisms which are present in extreme environments, like hot springs, for example, or very cold environment, they are called as archaea. And actinomycetes are also classified under bacteria now. Now they are called as Actino Bacteria. And these bacteria can be classified into two broad categories: Autochthonous bacteria and Zymogenous bacteria. Autochthonous bacteria are always present in the soil. They are the indigenous bacterial flora. So if you just go out from your college campus and then bring the soil now and look for the bacterial population and which are the diversity of bacteria present, and after about 6 months, if you go and take the sample, and the same bacteria will be present. That means they are the local indigenous bacteria. By mistake, one of the students adds a lot of urea in one particular spot. What happens? The Zymogenous bacteria, that is the urea decomposing organisms, develop in that particular spot. So when we isolate, we get these urea decomposing organisms. They are present only till urea is present. It is completely decomposed. After the urea is decomposed, they disappear. So they are referred to as Zymogenous bacteria. And what is the role played by bacteria in soil? They are nutrient cycling. Again, we all know about the nitrogen cycle, phosphorus cycle, and so on. They play a very important role in organic matter decomposition, production of industrially important secondary metabolites like enzymes, antibiotics, vitamins, and so on. Some of them cause diseases of plants and animals. I just included some of the diseases of plants. So when you go to the market, I do not know how many of you help your mother in the kitchen, but when you cut the potato, sometimes you see a brown ring in the potato. That is caused by the bacterium Ralstonia solanacearum. Even when you buy chips from the shop, you can see the brown ring. Okay, so that is the pathogen Ralstonia solanacearum. And bacterial blight of pomegranate, which is caused by Xanthomonas axonopodis pathovar punicae, is very serious pathogen attacking pomegranate. Our export market of pomegranate is very much objected or obstructed because of this particular disease. When the pomegranates are sent to some country, they are sending it back because of this bacterium. So the export market is very much affected by this particular pathogen. Some of these bacteria also cause animal diseases. Very common is anthrax in cattle, leptospirosis, and these are the animal diseases. And human diseases, all of us are familiar: typhoid, cholera, dysentery. They are all caused by TB, Mycobacterium, tuberculosis. All these are caused by by the bacterial pathogens. I just included a petri dish containing the different morphological colonies of bacteria. You can see variety of colonies: yellow pigmented, light pigmented, white, different shapes, and so on. And when we talk of morphology of bacteria, they are present in three main shapes. They are round shaped, which are referred to as coccus, or they are rod shaped when they are referred to as bacillus, or they are spiral shaped when they are referred to as spirilla. These are the three main shapes of bacteria. Coming to organisms, which are still smaller, the bacteria without a cell wall is referred to as mycoplasma. And mycoplasmas cause number of plant diseases. And you're all familiar with viruses. What are viruses? Viruses have a central nucleic acid, central part, either RNA or DNA, surrounded by a protein coat. And these viruses are also known to cause many varieties of diseases in humans, plants, and animals. What are viroids? Viroids are those where only the nucleic acid is present, there is no protein coat, and they also cause diseases. And opposite to that are the prions, where only the protein coat is present, RNA is absent, but they cause diseases. So all these organisms exist in soil. So that is the importance. And this I have already covered. Coming to population dynamics or population pyramid of soil organisms, as I say that if the organism is big in size, their number is less. If the organism is smaller in size, then the number is more. But studies that have been done so far has revealed two important correlations. What are they? There is no correlation between population density and importance to biological activity in soil. Okay, you got the point? There is no correlation between population density and importance to biological activity in a soil. In a particular volume of soil, let us say there are only 2 earthworms, but it has got billions of protozoa. So the number of protozoa is more, but when we talk of biological activity, the 2 earthworms are more beneficial compared to billions of protozoa. That is what we mean by no correlation between the population density and importance to biological activity. And again, there is no correlation between the size of the organism and the biomass they contribute to the soil. Okay. And here you can see the size of microbes are very small, bacteria and fungi, but they contribute 80% of the soil biomass. The macrofauna are much bigger, like which includes spotworms, earthworms, but they contribute only 14%. Microfauna and mesofauna contribute only to 2%. So smaller the size, but there is no relationship to the extent they contribute to the biomass that is present in soil. It's very easy to find out the biomass. There is a very simple method called chloroform method, by which we can determine the quantity of the biomass present in soil. And coming to the essential ecosystem services provided by soil biota. Here we can see what are the various services these soil organisms give to you and me, the mankind. Okay, the decomposition of organic matter. We know that the Nobel laureate Selman Waksman, what he said, if there would be no microbes or which can decompose organic matter, that is the dead animals, plants, etc., there will be no place for man to live. What he said is true. If all these animals and plants do not decompose, there won't be any place for us to live. Another important ecosystem service is regulation of plant nutrients and uptake. Then other one is suppression of pests and diseases. Okay, and so they biological control of pests and plant diseases. And nutrient uptake, the previous one, all of you are aware about the nitrogen fixation, phosphorus mobilization, and so on. And another important ecosystem, they contribute to the soil structure and regulation of the hydrological processes taking place in soil. So soil structure is very, very important, and the microbes and the soil fauna and flora contribute to this. And they also help in the carbon sequestration, which is another important topic given a lot of importance nowadays by funding agencies. Carbon sequestration, the soil, fauna and flora contribute a great extent, to the extent of they can fix atmospheric carbon in soil. Soil detoxification. We add so much of pesticides to the soil. fungicides, insecticides, herbicides. Some of them are very, very slow decomposers, degraders. So here again, the organisms, soil microbes play a very important role in degrading these toxic compounds, and which are commonly referred to as xenobiotics. And plant growth control, many of them help in the plant growth. As we have said that mycorrhiza plays a very important role. Similarly, the nitrogen fixing organisms and so on. Then some of them play an important role in the pollination of horticultural crops. So there are some insects, for example, Piponopsis pruinosa. This is insect which plays a very important role in the pollination of horticultural crops. So these are all the various ecosystem services provided by soil biota. The same thing has been given in the form of a picture here. So what are the various ecosystem services? They play a very important role in plant growth production, soil structure maintenance, nutrient cycling, biological population regulation, carbon cycling, decomposition of organic matter, water supply, climate regulation, and habitat support, and cultural value, and the biotechnology. When we say biotechnology, the biotechnologies come to soil for isolating organisms which can produce newer antibiotics, newer enzymes, vitamins, and so on. All these organisms come from soil. So the Millennium Ecosystem Assessment, which is the global organization which gives this assessment, makes this assessment, defines ecosystem services or the benefits people obtain from the ecosystem. Okay, they are the benefits people obtain from the ecosystem are covered under the ecosystem services, and they have been categorized into 4 different groups. What are these 4 categories? One is supporting services. The supporting services include nutrient cycling, soil formation from rocks, and so on. Second one is the provisioning services like food, fuel, fresh water. These are the provisions which the organisms give, because that we use. And the third one is the regulatory services like waste purification, climate regulation, etc. All they play a very important role. The last category of service provided by these microbes are the fauna and flora in soil, or the cultural services like the recreational aesthetics, the beautiful parks, everything we go and recreate ourselves. So these are the 4 categories of the services provided by the ecosystem, and the soil organisms provide all these 4 categories of services. Okay, all the services which are beneficial to mankind and the society. Let us take a couple of these examples, ecosystem services, and go into little detail and then see how they help mankind. Let us say organic matter decomposition. So the animals and plants, they are buried in soil. Okay, they undergo decomposition. What do these animals and plants contain? They contain sugars, starch, cellulose, pectins, proteins, fats, waxes, phenols, alkaloids, pigments, etc. And they are all acted by soil biota. Previously, microbiologists were thinking it's only microorganisms help in the decomposition of organic matter. Now we know it's just not the microbes. The soil fauna also plays an important role. So it is the soil organisms, flora and fauna, play an important role in the decomposition of organic matter. So let us take macrofauna. And macrofauna, like mollusks, millipedes, insect, they are the one which attacks the litter. Suppose let us say some leaves are buried in soil. So what attacks them first are these mollusks, millipedes, and insects. They puncture the leaf epidermis, and this process is referred to as fenestration. Next slide, I will show you the photograph of this. They puncture the epidermis, Thereby, they increase the surface area for the microbial attack. So, a very simple, interesting experiment has been done by a scientist. What will happen? What he has done is very simple. He has taken bags containing different sizes of pores. The pore size in the bag was different. So he took size of the bag with the pore size 7 millimeters. Size of the holes in the bag was 7 millimeters. And he added some leaves in it. And another treatment he had, bags with the pore size, hole size of the was only 0.5 millimeters. And he added lot of leaves and buried them in soil. After 3 months, he took the bags and studied to what extent it has undergone decomposition. Very simple experiment, but gave very interesting result. So found that when the pore size in the bag is 7 millimeters, which can allow many of the organisms, flora and fauna, the decomposition was 95%. When the pore size was very small, which will not allow macrofauna to enter, there was only 35% decomposition. So this is the leaf which has undergone fenestration. The epidermis has been eaten by the various fauna and which has a larger surface area for microbial attack. So as I told, when the pore size of the bag was 7 millimeters, the organisms, all microorganisms, invertebrates could enter. When the pore size was only 0.5 millimeters, all microorganisms, mites, springtails, spotworms, and invertebrates could enter, but it prevented the macroflora. So what do they do when once they macerate the soil? It is the mollusks, white millipedes, earwigs. They macerate the litter and pulverize it. Again, another group of fauna, earthworms, insects, they transport the litter down to the bottom of the soil. And again, the ones which are below the certain depth of the soil, they further macerate it and mix it with the soil. So we have a good decomposed compost mixed with the soil. And here again, with all this decomposition process, some part of the material it does not undergo decomposition. See, we call it as alkali soluble substance. It does not undergo, it's water insoluble, but it undergoes alkali soluble substance. So we call it as humus. It is present in the soil and undergoes very, very, very slow decomposition. Okay, and thereby release the nutrients to the plant. So every soil will have small quantity of humus present in the soil. This is another very interesting experiment that has been done by the collaboration between microbiologists and zoologists. What they have done, they have taken 3 different earthworms, Lubricus rubellus, Rubricus terrestris, and Aporectodia trapezoides. So these are the 3 earthworms, and they had a control where no earthworm was there. And the top of the soil, they smeared a thick layer of the bacterium, the plant growth promoting rhizobacterium, Pseudomonas fluorescens, okay, on the top and of the soil. And then they allowed these 3 earthworms in the 3 treatments and the control without allowing earthworms. And then they studied to what depth this bacterium is taken down. So in the control soil, it went up to 10 centimeters. That is because of the irrigation, watering. And of these 3 earthworms, you can see here this particular Lubricus rubellus took it to 20 centimeters depth. But here, these 2 earthworms took it to 40 centimeters depth. But again, if you see these 2, this particular earthworm has the population of the PGPR is more. Suppose they would have gone up to 60 centimeters, this would have taken it up to 60 centimeters. So from this, what is concluded is that the earthworms play a very important role in taking the PGPR down into the soil layer, thereby helping the deep-rooted plants the benefits of the plant growth promoting rhizobacterium. So very interesting but simple experiment. Coming to another essential service provided by the soil organism is the symbiotic nitrogen fixation. We know that nitrogen is a major plant nutrient, very important for crop production, is present plenty in the atmosphere. Some bacteria fix atmospheric nitrogen, but plants cannot directly take this nitrogen from the atmosphere. But bacteria living in soil can fix this nitrogen and make it available for plant growth. So there are certain symbiotic nitrogen fixing organisms. All of you are familiar with legumes, and legumes have root nodules. And what is sitting inside the root nodule is this nitrogen fixing bacterium, Rhizobium. Okay, so these rhizobia which are present in the root nodules fix atmospheric nitrogen and make it available for leguminous growth. And we also have certain bacteria which form nodules not only in the root but also in the stem. So this is stem nodulating bacteria. An example is Azorhizobium colinodens. And I know in Tamil Nadu, there were a lot of paddy is grown and the farmers raise a green manure plant and then incorporate into the soil before transplanting paddy seedlings. So that is Susbania rostrata. And this particular legume forms root as well as stem nodules. That is why the farmers grow this green manure plant and then puddle it into the soil before transplanting seedlings of rice. And similarly, Casuarina trees also form nodules, but it is not rhizobium, but it is by an actinobacterium called Frankia, an actinomycete. And the size of these nodules are quite big as a table tennis ball, and they fix atmospheric nitrogen. And that is the reason why they grow. Casuarina grows in sandy soils and in the seashores and by the side of rivers and so on. And nearly 50% of the nitrogen fertilizer can be saved through inoculation with the symbiotic nitrogen fixing bacterium. I've just included the root system of a legume, and you can see these nodules in the roots which are pink in color. More the pink in color, we presume that it's a better nitrogen fixer because of the leg hemoglobin concentration, which is more. More the leg hemoglobin, more it is presumed that it's a better nitrogen fixer. This is the slide which shows the stem nodule bacterium. And so you can see that stem of Susbania rostrata having these nodules caused by Azorhizobium colinodens. This is the Casuarina root showing the nodules caused by Frankia species, which are quite big and fixing atmospheric nitrogen. And we can screen and then select the best rhizobium for inoculating a leguminous plant. So we can isolate the organism, screen it through laboratory tests, and we have certain laboratory tests, and then we can go to the microplot study and then finally go to the field and then select the best rhizobium for inoculating a particular legume. You can see the difference between uninoculated and the inoculated legume. And so far, a lot of studies have been done on nitrogen fixation in our country on various legumes. I've just included some of them, which have been done in different parts of our country. That is how you see the variation in the yield increase. In case of chickpea, that is Bengal gram, the increase in yield up from 5 to 20% have been reported. In case of cowpea, 10 to 15%. In case of pigeon pea, that is red gram, 4 to 30%. In mung bean, that is green gram, 2 to 25%. And in urud bean, that is the black gram, 4 to 25% have been reported. And coming to the next category, we talked so far about the symbiotic nitrogen fixation, where the bacterium forms a nodule and fixes atmospheric nitrogen, okay, in the roots. The second category is the associative nitrogen fixing organisms. The bacterium lives in the rhizosphere and it enters the epidermis, and some of them can go up to the cortex region, and they sit there and fix atmospheric nitrogen. Classical example is Azospirillum, which is a very good nitrogen fixing organism. Another example is Gluconoacetobacter diazotrophicus. which is a sugar-loving bacterium. We call them a saccharophilic organism. And from the name itself, you can guess on which plants they are associated, like sugar cane, sweet potato, sweet sorghum, and so on, which contains sugar. And they fix atmospheric nitrogen. And that they are the associative nitrogen fixing organisms. The third category of nitrogen fixers are the non-symbiotic or free-living nitrogen fixing organisms. They never enter the plant root. They colonize the rhizosphere and then fix atmospheric nitrogen, which is made available for the plant growth. So Azotobacter is a classical example. Lot of work has been done on Azotobacter, and it also produces growth promoting substances, and they are also antagonistic to some other plant pathogens. Another example of a non-symbiotic or a free-living nitrogen fixing organism are the cyanobacteria. You had such wonderful lecture by Professor Tajuddin on cyanobacteria, and these were earlier called as blue-green algae. They also fix atmospheric nitrogen. They add organic matter also to the field, and they produce also vitamins and auxins. And I just included a few tables of the response of crops to azospirillum inoculation. In case of amaranthus, a nearly 36% increase in yield has been reported. In coffee seedlings, up to 50% increase has been reported. In case of free-living nitrogen-fixing organism, Azotobacter, in cabbage up to 19%, tomato 28%, in onion up to 22%, and in brinjal up to 42%, and has been reported. In cyanobacteria, there's a lot of work done in Tamil Nadu, Madhya Pradesh, and Orissa. And here I can say that one example I've taken, up to 17% increase has been reported from Madhya Pradesh. And we know the heterosis, these cyanobacteria have heterosis, and these are the heterosis, the site of nitrogen fixation. And this is one slide which I took when I visited Kanyakumari district of Tamil Nadu. And here, the farmer himself is raising the cyanobacterial inoculum. It's very simple. You make a trench, put a plastic sheet, and then add some soil and water and inoculate with the cyanobacterium. In about 3 weeks' time, it covers the entire area, and this can be added, which can be collected and can be introduced in the paddy field at the time of transplanting rice pod. Otherwise, you can even dry it and then use it later whenever you want. So this is a very good, simple technology where the farmer himself can raise the cyanobacterial inoculum for introducing the paddy fields. Now, talking of the nitrogen cycle, after talking all this about nitrogen fixing bacteria, we know that nitrogen is present in the atmosphere, and which is the nitrogen fixation takes place by the nitrogen fixing bacterium, and they fix the nitrogen in the form of ammonical form of nitrogen, and most of the plants prefer nitrate form of nitrogen. So this ammonical form is again converted to nitrite by Nitrosomonas, which is again converted to nitrate by Nitrobacter, and this nitrate is taken by the plant roots. This is the nitrogen cycle. And there are some bacteria present in soil which are referred to as denitrifying bacteria, and these denitrifying bacteria act on nitrates and release the nitrogen back into the atmosphere. This is the type of nitrogen cycle we have. Coming to phosphorus, another important plant nutrient, tropical soils, as I told in the morning, tropical soils are deficient in phosphorus, and in addition to being deficient, they are also phosphorus P-fixing, up to 75 to 80% of them converted to unavailable form. But there are certain bacteria and fungi which can act on this phosphorus, unavailable form of phosphorus, through the production of organic acids, and then convert them and release the H2PO4 ions which the plant roots can take. So examples of these organisms are Bacillus polymyxa, Pseudomonas striata, Aspergillus avamori, Penicillium digitatum. And lot of work has been done by adding rock phosphate, which is a very slowly available phosphorus content, and mixing it with phosphate solubilizing organisms. Again, it could give the same amount of plant growth as that of adding a phosphatic fertilizer. Again, lot of studies have been done much in the earlier years, adding rock phosphate with phosphate solubilizing organisms and then comparing it with the application of soluble phosphatic fertilizer. And this is how we test the phosphate solubilizing ability in the laboratory using in Petri plates. We can introduce the tricalcium phosphate and then measure the zone of solubilization. The bigger zone means the better P solubilizer. I have included 2 organisms, one fungus and one bacterium, which are very, very efficient phosphate solubilizers. As I said earlier, a lot of studies have been done on phosphate solubilizing organisms in our soil. And again, increase in yield has been reported, 56% in cabbage, 37% in tomato, 43% in cucumber has been reported. Coming to arbuscular mycorrhizal fungi, I will skip this because I have already covered this in the morning. Then coming to the next category of organisms, the plant growth promoting rhizomicroorganisms. There are a lot of interest in this PGPR nowadays because they increase plant growth through production of phytohormones directly or indirectly by suppressing plant diseases. In a strict sense, nitrogen fixers, phosphate solubilizers are also PGPRs, but people usually consider them as separate. And when they talk of PGPR, they think only of certain bacteria like Pseudomonas fluorescens, Burkholderia, and so on. So these are some of the PGPRs: Pseudomonas fluorescens, Burkholderia, Methylobacterium, Azoarchus, Pantia, Pennibacillus. And globally, most of the work has been done on Pseudomonas fluorescens, followed by Burkholderia cepacia. And to some extent, work has been done on Methylobacterium also, especially in Tamil Nadu. Quite a bit of work has been done on Methylobacterium. Other organisms, not much has been done. So we had a project funded by DBT to work on these organisms which have been less worked. And the work was done with French bean. And here you can see different PGPR were used. The one which was the best was Pantia agglomerans, was the best, increasing the fruit yield as well as the plant dry weight. So from the lab, we took it to the glasshouse, finally to the field, and finally in the farmer's field. So it was working extremely well. And coming to another group of organisms which play an important role in the biocontrol, we can say that microbes play a very, very important in the biocontrol of plant diseases. And if you see these biocontrol agents which has been worked on, these are the various crops, various diseases, and you can see which is the biocontrol that has been used agent. Mainly it is trichoderma. So trichoderma is a very effective biocontrol agent which can help in the biocontrol of various soil-borne plant pathogens. And just including something about insect pathogenic organisms, again, we can have Bacillus thuringiensis, very effective in controlling the insect. And you can see the growth of Bacillus thuringiensis in the plate and the dead larva here. And microbes also play a very important role, soil fauna, in the pesticide degradation. As I told that some pesticides, and in fact, some of these microbes use the pesticides as a source of carbon or energy and thereby make them to disappear after a few years. And the extension to the other fields, there are several soil fauna and flora which are used, mainly microbes. Several actinomycetes are used for the production of antibiotics. We know streptomycin is used, a variety of penicillin is used, and the vitamins, vitamin B12, thiamine, and here the cytokinin, gibberellic acid is also produced by Gibberella physicora, and proteolytic enzymes are produced by Mortierella. So several compounds are produced by these organisms. And what is the value, economic value, given to these various ecosystem services provided by these organisms? Unfortunately, we do not have much information from India, but American scientists have done this job. And for example, organic matter decomposition, they have given a value of $760 billion per year. For nitrogen fixation, they have given $90 billion per year. For biocontrol of pests, they are given $160 billion per year. So this type of work is needed in our country, where we can as microbiologists, we can collaborate with the economists. We tried in one of the projects to do this, but we were not very successful. What are all the threats to soil biodiversity? The main threat is the mankind because of the industrial pollution. So you can see mainly pollution, and then the agricultural practices. Earlier we were following mixed farming, now it is monocropping, and now climate change, overgrazing by cattle, introduction of alien species, soil erosion, pollution to the soil, land degradation, desertification, fire management. We all talk about the fire that is given in Punjab to the rice and wheat in a straw. All these are there, which are threat to soil biodiversity. They reduce the diversity and the population in the soil. Again, the same thing in a different way it presented. The human intensive exploitation is the main reason threatening soil biodiversity. How can we conserve soil biodiversity? So these are the measures that are suggested. And one is the various soil amendments, organic amendments, less tillage. I won't say no tillage, less tillage. Diversification of cropland, go for mixed cropping rather than monocropping. Fire management, soil erosion control, agroforestry, have some trees as the African scientists follow, and prevention and restoration of invaded sites. So these are the methods which are suggested for conservation of soil biodiversity. This is a very interesting slide. I am sure that all of you will enjoy this slide. Children love to play with soil. As a small boy, I think I have also played throwing soil on each other. And so they enjoy playing with soil. But to what extent did they know about the biodiversity in soil is a question mark. So people have solved this problem. In a primary school, a teacher asked the children to draw what they know about the biodiversity in the soil. This is what a primary school child has written. You can see that it has written so many living organisms present in soil. That means the soil, the child knows something about the living things in soil. Okay, this is the primary school child. Now, this is another thing which has come from the school with the smaller children and the grown-up children. So 4 to 7 year old children, when they were asked about soil biodiversity, what did they say? One child said, without soil, there is only a big hole. Another child said, trees grow on soil. Another child said, my ball won't bounce back. Another child said, without soil, we will tumble down, go down. Little grown-up children, 7 to 9 years, what the one child said, plants grow in soil. True. In soil, there is seed. I don't know if my child must have seen a place where a lot of seeds were fallen down from some tree or something. So that is what it has said. And another child said, foxes lives in cave in the soil. I don't know whether it saw a bandicoot or some rat, but it thought it is a fox. Foxes live in soil. And the bigger children, 9 to 11-year-old child, one child said, trees grow in soil. True. And it went one step further. What did it say? They produce oxygen for humans and animals. So that means the child knows about the importance of plants. And another child said, in soil, I can find earthworms that I need to catch fish. Okay, so that means our children know something about soil biodiversity, but it is our duty to educate them further. Okay, United Nations declared 2010 as the International Year of Biodiversity, and the European Atlas of Soil Biodiversity was brought by the European Commission in 2010. So that was the first soil biodiversity atlas, and that is only for the European countries. And the FAO organized a meeting and an international workshop with the title Managing Living Soil in 2012 in Rome. What came out of this meeting is a program called Global Soil Partnership Program, where they wanted to bring out the importance of soil to the common public, like how the soil plays an important role in food security, in mitigation of climate change, poverty alleviation, and sustainable development. And an international year of soil was also framed. And what is the purpose of the International Year of Soil was to create awareness among the people. And out of this Global Soil Partnership Program, 5 pillars were recognized. The 5 pillars are: one is soil management, second is investment, third is research, information and data is the fourth one, and fifth is the standard. So I feel that pillar 1, dealing with managing living soils and research in soil, are very important. And I was very fortunate to be a member of the committee of both these pillars. And United Nations also declared 2015 as the International Year of Soils. And again, December 5th of every year is declared as the World Soil Day. I am sure that all of you are familiar with the Mother's Day, Father's Day, Valentine's Day, which went very few days back. And I, at least in future, remember the World Soil Day, which is 5th December. Okay. And we know that the Global Atlas on the European Atlas on Soil Biodiversity was brought earlier. But later, in 2013, FAO wanted to bring a Global Atlas on Soil Biodiversity. So nearly 100 scientists from 26 countries were invited to contribute to this atlas. For example, if you take nitrogen fixation, nitrogen fixing bacteria, 3 to 4 scientists from 3 to 4 different countries were asked to work on this and come with the write-up and the photograph for nitrogen fixing organism. Similarly, another 3 to 4 scientists on earthworms, like that. And so this particular atlas, what they said, it is not for scientists, it is for common man. So the language should be very simple. A common man must be able to follow, but it should have beautiful photographs. That was the criteria. And this was completed, and this was brought, the Global Atlas on Soil Biodiversity was released in 2016 at Belgium, in Brussels. And I was very fortunate to, I was invited to contribute to mycorrhizal fungi. And I would like to add, I was the only scientist from India invited to contribute to this Global Atlas on Soil Biodiversity. I have just included here the front page of the Global Soil Biodiversity Atlas and the last page. So you can go to the FAO website and then type Global Soil Biodiversity Atlas. You can freely download the atlas. Or if your library wants a hard copy, it costs 26 euros at a subsidized rate. So you can write to FAO, then they will be able to supply the hard copy. So finally, before closing, I just want to tell 1 or 2 slides of the importance. We always, what do we say? When the child is trying to play in soil, we say, don't play in soil, it is dirt, something. That is how we prevent our children from playing in soil. But this is one of the very recent reports which came from Czechoslovakia. The children playing in soil, the children of gardeners, they were found to be more happy than the city children. The reason, they were trying to analyze what's the reason. And they found that the particular bacterium, Mycobacterium vaccae, which is a saprophytic bacterium present in soil, was entering the children either through inhalation or through cuts in their body. And this particular bacterium produces a hormone called as happy hormone. The name of the hormone is serotonin. So, which is an antidepressant, and it decreases anxiety, makes you feel very happy and relaxed. So that is how they found the village children playing with soil were more happy compared to the city soils, where children, where they have more problems when and all the transport and pollution and so on. And this was later confirmed by another work that was done in Finland. The National Institute for Health and Welfare in Finland, what they found was the children in rural areas had only 4% asthma, whereas in case of urban children, the incidence was 29%. They were trying to understand this, and they came to the conclusion the rural children were playing with soil and they were exposed to soil with beneficial microbes, which increased their immunity to asthma, whereas the urban children were exposed to traffic and pollution and had no chance of playing with the soil. So this was confirmed by later studies in other parts of Europe and USA. The conclusion from this is early life exposure to soil matters a lot, increasing the immunity in children. So let us not prevent the children from playing in soil, at least in future. What we can draw as conclusion: soil is the reservoir of several organisms. Soil biologists study them in isolation. Soil biodiversity is critical for plant growth, climate regulation, ecosystem functioning, disease control, and human health. There is need for collaboration among soil biologists working in different groups of soil organisms. I would like to emphasize this point very much. We, as microbiologists, will work only with microorganisms. We will never interact with a zoologist or an entomologist or a soil scientist. I think there should be collaboration. Unless, when we collaborated in my lab, I'm telling my own experience, when we collaborated with the zoologist group and the entomology group, we came that earthworms and wasps play a very important role in dissemination of fungi. And again, I see in some places, I'm very sorry to say this, when I go to the microbiology department itself, people working on fungi don't see the face of people working on bacteria. So these things should go away. There are coordinated studies also needed between soil biologists, soil chemists, agronomists, ecologists, and name it, whatever it is. And more research is needed to understand the complicated and interlocked processes carried out by organisms in soil in order to derive the most benefit from these soil organisms for mankind. So healthy soils, we have healthy plants. When we have healthy plants, healthy children and healthy adults. Thank you very much. If you have any questions, I'll be happy to answer. Any questions? I'll be happy to answer. Yeah.
Thank you, sir. Thank you for your enlightened lecture. We will next move on to the questions session.
Yeah, I can put in the chat box. That'll be easy to see and answer. Yeah. Yeah. Since 2 o'clock. Okay. We did that. The one that the previous one, I think. Yeah. And this is Mycorrhiza. Yeah. Yeah. So I see one question here. With human impacts. One presentation. Anyone? Okay. Question mycorrhizal plants. That also I answered from here.
Is azolla a symbiotic nitrogen fixer?
Which is
type of growth is shown by phosphate solubilizing bacteria, PS, biofertilizers?
Pardon?
Is azolla a symbiotic nitrogen fixer? Which type of growth is shown by phosphate solubilizing bacteria, biofertilizers?
Yeah, azolla is a water fern and which harbors anabina or anabina in the plant leaf system, and thereby it fixes atmospheric nitrogen. So when a farmer adds azolla to the rice field, it not only adds lot of biomass, Okay, organic matter plus nitrogen before transplanting rice seedlings. This is a practice that is followed by the rice growers. Okay, I see one more. Which type of growth is shown by phosphate solubilizing bacteria? Like any other organism, if we are working with a particular phosphate solubilizing actin, there is no special growth, just like any other bacterial growth. Only thing is, when we try to test it, then we try to test it on a tricalcium phosphate medium where we can measure the zone of solubilization. And another question, you have explained points about regulatory services of soil microbes and climate regulation. Do soil microbes can regulate global warming? Yeah, I think more studies are needed in this particular area. Just a beginning has been made. More studies are needed in this particular area. Yeah, but global warming alters the organisms in soil. Okay, then thank you. All thanks. Okay. Any other question?
Thank you, sir. Thank you for your presence with us, even though you are in that medical emergency to take care about daily, but you interact with us, our students and external participants. We wholeheartedly thank you for addressing various basic questions and some research-based questions. I wholeheartedly thank you, sir, for being a convener of this lecture workshop, for mentoring me and support me and guiding me in various platforms. I thank you, sir, for you are the only sole responsibility for the third year we are conducting this lecture workshop, sir, with your blessings and leadership. I wholeheartedly thank you for the initiative taken by you for the betterment of the students and researchers, sir. Thank you.
Oh, thank you very much, Dr. Riaz.
Thank you, sir. Sir, and now we have with the formal vote of thanks by our UG head and PhD head, Dr. Jayaprakash. Sir, please proceed, sir.
Okay, sir. Thank you. So I take this opportunity to thank our speaker of the session, Dr. D.J. Bhagyaraj, for his elaborate lecture on Arbuscular Mycorrhizal Fungi for sustainable agriculture and soil biodiversity. Thank you, sir, for your enlightened lecture, and we have that more researchers will be delighted to work in AM fungi in near future. In a special way, we, the faculty members and students of Symbiotic College, pray for your good health, sir. Thank you very much, sir, for your enlightened lecture, and we also pray for your good health. Also thank the participants for your patient hearing and questions. With this technical session 4 ends, and we shall meet tomorrow at 9:30 AM for day 2 of the workshop. Thank you all and have a pleasant day ahead.
Okay.
Sir, thank you.